Trogenix is a clinical stage precision genetic medicine company. Potential for curative responses in solid tumours, leveraging our Odysseus platform, AI-guided, functional biology-driven discovery engine that allows us to discover universal vulnerabilities of the disease cell state and develop therapeutics that target disease at its source. We're in the clinic with our lead program in glioblastoma, phase 1/2 UK, US, follow-on programs in colorectal liver mets, liver cancer, and non-small cell lung cancer, and an earlier pipeline across oncology and regenerative medicine. We have a leadership team that spans innovation through to commercialization. Steve is a world leader in stem cell and cancer biology, our founding CSO. Myself and Juanma have a career story of developing precision genetic medicines to the clinic, and in Atif and Tay we have a track record of execution through the clinic and into commercialization. We're at the beginning of a cancer care revolution. We've seen terrific advances with targeted agents for cellular pathways or surface antigens, giving patients significant extensions to life. But we're only at the start of that journey. Too often we've only been able to treat subsets of patients. And often if you're only going after a singular target, then providing that true differentiation between the disease cell state and the healthy tissue is difficult. We're often compromising potency to balance the off-target side effects. And then we're often undermined by the plasticity of the cell, so we're only seeing extensions to life. What if we go after this cell? The cell is a fundamental unit of cancer. It's characterized by a set of master transcriptional factors and cofactors that define that cellular identity across all patients. Trogenix has harnessed that to create a genetic switch that expresses combination payloads from within the cell. No off-target side effects means we have no compromise, so we can hit the disease cell hard and early. And because we're going after that complex network, it's incredibly hard to design around. You'd have to break the identity of what the disease is. So through our Odysseus platform, we've uncovered that master transcriptional network that describes the disease cell state. We harness that and create synthetic superenhancers, genetic switches, that you can think of a little bit like a QR code. So a QR code is very precise, but it has built-in redundancy. It can be obscured, it can be damaged, it still reads out precisely. The SSE reads out against that complex network of master transcriptional factors, cofactors, with built-in redundancy, so we can manage the plasticity of the disease. With that precision, we can really begin to think about changing the paradigm in cancer care from chronic disease management to potential one-and-done curative therapies. We use intratumoral delivery of an AAV and express combination payloads, so a kill and a cytokine, to give us an innate and adaptive immune response for lasting immune memory. We finally have the tools to address some of the most desperate of unmet needs. Our lead program is in glioblastoma. Most patients won't make it 12 months. The starting point, we develop our SSE. We built the world's biggest biobank of patient-derived cell lines, 58 patients across the heterogeneity of the disease, fully characterized. And you can see here, no, you can't. You can see here the complex of the genetics. But at the phenotypic level, they all share the same master transcriptional network defining the disease. And that's what we're harnessing, so we can think about treating every patient. We have our lead candidates. We then are testing it directly in patient tissue. So we're based on the hospital campus. This is human tissue straight from surgery, straight into the lab. Not frozen, it's straight into the lab. And we've developed techniques to keep it alive for over a week. Okay, so we can see here, this is the tumor cells in red light up. Importantly, the macrophage, so 50% of a brain tumor is the macrophage. they're left untouched, and we need them. They've been corrupted. They form that protective tumour suppressive microenvironment. They're trying to protect it. They've been corrupted to try and protect the cancer cell. So we need them to be re-educated and clean up the mess. But the tumour itself, the surgeon can often resect it, so it's not that will kill you, it's the infiltrating tumour cells. So in surgery, the surgeon will remove the tumour, but they'll also remove as much of that healthy margin as they can, because they know within 6 months that tumour will rebound, and then the patient will die. And look at this, for the first time, we have the tools to pick out individual infiltrating tumour cells, leaving the neurons, the astrocytes, all the good stuff untouched. And this isn't just one patient, this is every patient. Okay, so that, across the full spectrum of genetics, so the IDH mutants, the wild type, even grade 3 and the recurrent patients. And in fact, if you think here, look, at grade 3, the less aggressive cancer, you can see that we have less activity. That the recurrent patient, where typically the tumour is more aggressive, we get a stronger response. So it's a tuned therapeutic. Okay, so we have the selectivity. We spent 5 years building a meaningful syngeneic mouse model. It was published in Cell a few years ago. It was actually instrumental in helping us understand that corruption of the macrophage and that highly suppressive tumour microenvironment, highly aggressive infiltrating cells. When we dose, when we dose, that tumour is already really significant. That mouse will die in a couple of weeks. Within 1 week, it begins to clear. Within 4 weeks, it's gone. And it stays gone, and it's not just one mouse, it's pretty much every mouse we're treating. They'll have a full 12 months to the end of the experiment, no weight loss, no neurodeficit, no tox. What's more, when we come back and re-challenge those mice at 6 months, we inject another 200,000 tumours into the opposite hemisphere. The tumour won't take. We see a lasting systemic immune memory. With that precision, with those types of results, we need to go into the clinic. And we have a terrific safety profile. Maybe just to mention part of it, which is the kill and the cytokine are on the same transcript. That means our cytokine, IL-12, can never get out of control. We have a pulse of IL-12, and then we kill the cell. So it's an almost biophysical pulse. And in fact, we can actually see that happening on those tissue slices I was showing you. Because of that precision, both the MHRA and the FDA are supportive of going ahead of standard of care. Now, this is really important, right? It's an immunotherapy. You want to treat people before they've been ruined by chemoradiotherapy. What's more is that we dose, and then 3 weeks later, we'll resect the tumour. That means we've got what's left of that tumour in our hands to interrogate for expression of the transgenes, the kicking in of the immune response, so the Th1, interferon gamma. That's an incredibly informed position to run the trial and really propels it forward. To switch track, I wanted to mention briefly the underlying platform and touch on some of the pipeline. So the Odysseus platform, AI guided. Now, I use that with a little bit of caution. I've been working in machine learning for 10 plus years. It's interesting to see how the word's bandied around so often now. The really important thing for me is the integration of our bioinformatics, our machine learning, with our synthetic biology tools, with our meaningful functional biology screening. So Odysseus has a proprietary front-end data lake with public and private data that's curated, QC'd, qualified, prioritized. From there, we can rapidly identify our elements and screen hundreds of SSEs, payloads, and in the context of the full vector, so that the final drug candidate screening. So we can build hundreds within a week and test it on primary cell lines, patient tissue, meaningful syngeneic models. With that, we can create drug products for every disease cell state. We focused on oncology and regen med. We're in the clinic with glioblastoma, IND-enabling studies for diffuse midline glioma, so pediatric disease, colorectal liver mets and liver cancer, and then follow-on programs in oncology and regen med. So to finish up, a slide each on some of our pipeline. So this is the same idea. This is fresh patient tissue from colorectal liver mets, straight from surgery into the lab. We light up the tumour. We don't see any off-target tox. Same story in liver cancer and in non-small cell lung cancer, both in adeno and in squamous. And we're even now able to see in a subset of fibroblasts precise activation of SSEs. I will finish there and happy to take questions. Thank you.